Good evening. I don't know if we lost a few more or what, but maybe we'll get a few more coming. I think Miss Pat, would you mind opening us up in prayer, please, ma'am? Appreciate it. Please.
Lord, we come to you with thankful hearts that we are able to learn from you, from Lauren, from his study. We thank you for his study, Lord, and that the truth that he reveals through you, through your word. This is an exciting study, Lord. And we thank you that we are blessed with this opportunity to learn. We thank you for your scripture. We thank you for this beautiful day and for your many, many blessings. Be with us now, Lord, as we study your word. Amen.
Amen. Thank you. All right. Well, tonight, if you've been on the fence at all about whether or not we are a creation of God, I think we'll be able to dispel all of that tonight. It's interesting. Yesterday, I guess earlier last week, maybe the day before yesterday, James had called or texted me and asked me if I would read out of 1 Peter 5. And normally when he asked me to do that, I'll actually go look at some commentaries on what we're about to read. And one of those is Grace to You, John MacArthur. I don't know if y'all see that, but you can look at sermon series and whatnot. I looked up on 1 Peter chapter 5, and we were looking at 10 through the end. I thought this was pretty applicable to what we've been studying. I'm just going to read what he comments. It's a little bit long, but let me read it. He's talking about the ways the mindset of society is today, and even some of the churches, the mindset of some of the churches. But he says these kinds of things have produced a sort of mindless Christianity that is in fact the antithesis of everything that God has designed in his church. God never intended that his people would somehow connect up with him and worship him without the use of their minds and without basing their relationship on a very clear and precise understanding of truth. In Psalm 32, verse 8, the scripture says, I will instruct you and teach you in the way in which you should go. I will counsel you with my eye upon you. And then it says, do not be as the horse or the mule not having understanding. There's no virtue in not understanding. There is no virtue in lack of information. There is no premium on not knowing. In Psalm 73, verse 22, the psalmist writes, when my heart is embittered and I was senseless and ignorant, I was like the beast before you. Senselessness and ignorance before God is considered to be that which is characteristic of an animal and not of a man. In the prophecy of Isaiah chapter 1, verse 18, come, let us reason together, says the Lord. In Hosea the prophet in chapter 4, what would sum up the very important truism, my people are destroyed for lack of knowledge. God has never put a premium on mindlessness. It's just the opposite. I agree with him. It's kind of what Kaylee McEnany, I had that slide up a couple of weeks ago, that her take when she was talking about the death of Ravi Zacharias, that we as Christians don't have to leave our brain at home, that we can actually see the world around us and think our way through this issue of creation and the primeval accounts. So last week I gave you some homework. It was to tell me what information was. Anybody come up with an answer? Thank you, Adam, by the way. I watched all 45 minutes of that. It was phenomenal. I agree with every bit of it. Nobody did their homework, did you? You all failed. What is information? Anybody got an idea? You just don't want to be called out, do you? I'll give you my idea of information. There's something that I know, tacit information that only I know, and I want to be able to convey it. Now, last week I put up on the screen a bunch of gibberish, and you couldn't figure out what it said. Now, it had all the right letters, but it didn't convey any information. It's when I have tacit information between my ears and I use some medium, whether I write it, whether it's audible, whether it's in any other format, and it comes to you, and you are able to interpret it along a particular protocol, English language, English characters and writing, and now that thought that I had is now between your ears. That's probably a pretty good definition of information. But in information technology and information science, there's actually a couple of different kinds of information. There's what's called Shannon information, which is pretty much data points. If I was to take a Scrabble game with all the letter pieces and I just threw it down on the ground, they would be in some random order. And you could say there's an A over there, there's an M over there, and there's a Z over there. These are points of data, but they don't mean anything. But if all of a sudden they just dropped on the floor and said, "Hello, Lauren," I might think otherwise, right? So you've got information that is Shannon information, which are mere data points, and then you have informational or functional data, which is useful for knowledge. You're conveying something. And then you have what's called prescriptive knowledge, which is kind of like grandma's cookbook. It'll tell you how to do something. Tonight I want to walk through some of the later two, functional and prescriptive. So we're going to be looking tonight at, I'm telling you, you're going to, if you've not seen these motor proteins and nano machines, you're in for a treat tonight. Go to the next slide. So Dr. Stephen Meyer, he wrote two really good books. I know Adam says he's read both. Darwin's Doubt, I kind of called it the other day Darwin's Dilemma, because he had more to it than what he realized that he was messing up. He also wrote Signature in the Cell, where you can see what I would call the fingerprints of God in the cell. And what he says in multiple interviews and in both of these books is that biology is an information system. It's an information transmission, storage, and processing system. In his book here, Darwin's Doubt, the animal forms that arose in that Cambrian layer. Now you remember that Cambrian layer, if we dig down to a point, there's a strata where all of a sudden all those animals appear fully formed, fully recognizable, irreducibly complex, a whole variety of life that we find in the strata. And when you go down below it, the Precambrian, they call it Precambrian because there's nothing else there. That explosion of life just all of a sudden happened. So these animals that arose in that Cambrian layer not only did so without clear material antecedent, in other words, they didn't have these primordial life forms or transitional life forms, but they came on the scene complete with digital code, dynamically expressed integrated circuitry, and multi-layered hierarchical organized information storage and processing systems. Well, what the heck is he talking about? Robots? Last week I tried to explain a joke that only information scientists will really understand. Go to the next one. That's computer nerds will immediately get that, and it only works if you read it yourself. Because most of us will say there are 10 types, and us computer geeks see that as 2 types of people in the world, those that know binary, those that don't. I put a cheat below it, ones and zeros. So I said last week that all the world's information we can store on computer systems, and computer systems are either on or off. They have a 1 or a 0 that represents the kind of data, and we have a particular protocol, and I'll explain it here in just a moment. Go to the next slide for me. We in the English language have 26 letters. We talked about this a little bit last week. That we can take all the world's information. We can take the Encyclopedia Britannica and we can communicate it. You can read it. We can take the thoughts of the authors, the various authors, and communicate that to you so that you can put that between your ears, and now you have the same tacit information that they had. So we can do that with the English alphabet with 26 characters. Go to the next. In DNA, we have what are called nucleotide bases. We represent those with A, T, G, and C. So instead of 26 characters, or only 2 in the case of computer systems, I just said a moment ago, we can store all the world's information with just two letters, two numbers, the binary system, if we use the right protocol and use it in the right way. Here we've got 4, A, T, G, and C, that are the nucleotide bases. Now, in computer systems, go ahead and go to the next slide there for me, James. In computer systems, the way it works is we have a 5-volt DC voltage, which represents a 1, or we have 0 volts, which is 0. And if you've ever stuck your finger in a light socket, you know that there is a physical response, there's a physical attribute to having electricity. There's an electrostatic charge that is at 5 volts. And the way we have this protocol, you can see that sine wave, basically a digital wave up at the top. And that actually spells out the letter W in ASCII. I'm not going to get into the whys and the wherefores, but it goes 0, 1, 0, 1, 0, 111. And we can see that W being that binary protocol. 8 bits, the bits are the letters, 0 or 1, and you group them together in 8 bits, you make a byte. And that's a word. And you string these words together, and all of a sudden we can start making the computer do things, such as print out the word word. We can see the same thing in genetics. In DNA, rather than an electrostatic response, we have a chemical response. And the body responds to the chemicals in unique ways, which we're going to look at closely tonight. Hit the next slide. So, Darwin, in his madness on the origin of species, he had a theory, and he said toward the back of the book, if it could be demonstrated that any complex organ existed, which over the period of billions of years and millions of years of natural selection and macroevolution, if it could not possibly have been formed in that way, then my theory would absolutely break down. And so we've got a choice. You've got Darwin's theory of evolution, which, by the way, today they call it neo-Darwinian evolutionary theory. And the reason it's neo is because he got it wrong and they know it. And now they're trying to adapt it in new ways that kind of make sense with the science that they misinterpret today, in my opinion. The other option is what I said in session 2. We would hang our hat on Genesis 2:7, that the Lord God formed man of the dust of the earth. He breathed his neshama, the breath of life, into his nostrils. And in an instant, he became an irreducibly complex, interconnected, interdependent system of systems. He became a living creature. Go to the next slide. And by the way, that is true of all life. You can see this in Ecclesiastes. For what happens to the children of man is happens to the beasts. It's all the same. As one dies, so the other dies. They've all had the same breath, that neshama, that breath of life. And man has no advantage over the beast, for all is vanity. All go to one place, all are from the dust, and to dust they will all return. Creatures, whether man or beast, have the breath of life, and and we're talking specifically about land animals that we would have had on the ark. So, for Mr. Darwin, if you go to the next slide, his theory is on the left that we've seen this before. There would have been transitional life forms that went back to eventually, if we go down deep enough and long enough and search thoroughly enough, that we would find, he actually says in the back of his books, we should in a period of time be able to find 3, maybe 4 transitional life forms that actually got us to that single primordial life form that all life came from. Of course, that never happened. It's been 160 years since he had that theory. And what we find instead is on the right, the actual fossil evidence of these irreducibly complex system of interconnected and interdependent systems. Living creatures, fully formed, variety and the speciation of life, all in a Cambrian explosion, all at the same layer of life. Let's look at what that life looks like. Go to the next slide. Y'all remember COVID? Hard to forget, right? I said back in February it was you could love your neighbor as you'd love yourself as long as you did it 6 feet away. COVID. October 10th of 2021. This is 2 months after I started at the seminary and several months before I decided to do it on this my paper on this topic. But I happened to be looking at the mRNA technology that they use for COVID vaccines. You all remember the big three. You had Moderna, Pfizer, and Johnson & Johnson were giving the COVID vaccines that were on the market. And all three of them were based on mRNA technology. Now, I don't expect you to read what I have highlighted over there, but I was researching it and I happened to look at the Moderna website. And so I'm going to blow it up big for you. By the way, this paper this information, that page is no longer available. They've taken it down and it now says some madness about genetics, and it's been around for billions of years is what it now says. But they do not have this anymore. I screen captured it at the time because the argument that they gave, that the scientists at Moderna in their mRNA explanation, what they gave for the evidence for God's creation was just astounding to me. And I'll just read it to you. Without mRNA, your genetic code, I'm a software engineer by education. I did professional software development for years. It fascinates me when I see something like your genetic code, but your genetic code would never get used by your body. Proteins would never get made and your body wouldn't, actually it couldn't, their words, not mine, perform its functions. We need to talk about messenger here in just a moment, but messenger ribonucleic acid, or mRNA for short, plays a vital role in human biology, specifically in the process known as protein synthesis. They're developing, making, building proteins. mRNA is a single-stranded molecule that carries a genetic code, there's that word again, from DNA in the cell's nucleus to the ribosomes, the cell's protein-making what? That's a fascinating word. What the heck do they mean by that? Go to the next slide. Transcription was the second half of that. Through a process known as transcription, an RNA copy of the DNA sequence for creating a given protein is made. A copy. This copy of messenger RNA travels from the nucleus of the cell to the part of the cell known as the cytoplasm. That is what Darwin in that rudimentary microscope looked and saw the cell, and he saw that translucent, ooey gooey chewy center of the cell that he said was jelly in the middle of the cell. Without knowing anything about the cell, he saw that the center, that was the cytoplasm that he was seeing and calling jelly. So that's a part of the cell known as cytoplasm, which houses the ribosomes. Ribosomes are complex machinery in the cells that are responsible for making proteins. And then through another process known as translations, the ribosomes, it's their quotes, not mine, the ribosomes read the mRNA and follow the instructions, creating the protein step by step. Remember I said that there is Shannon information, there was the functional information and prescriptive information. Prescriptive information tells you not just some piece of information, but tells you how to do something, like Grandma's cookbook. Inside of our DNA, particularly for building a protein, we have a blueprint. It's a chemically laden blueprint that is spelled out in 4 characters, A, G, C, and T. And it reads this, and we're going to see how it works. We've got some animations that I want to show you tonight, but I'm going to give it to you this way. Go to the next slide. Basically, when you need a cell, you've been in the gym, you've been working out, you're tearing down your muscle, the inside of your cell, the nucleus, basically needs a protein to be built. And it sends, let's just call it an email. It's a message. And it basically says this in 4-digit code. It's a string, a series of genetic codes in these nucleotide bases. Dear ribosome, please make me a bicep muscle protein to make the bicep protein, do the following. Step 1, do this. Step 2, do that. Step 3, do something else. And then when you're done, I want you to deliver it right here to my bicep muscle. It actually puts in the DNA addressing information so it knows where to put the protein muscle once it's been built. So what the scientists at Moderna, Pfizer, and Johnson & Johnson did is they hijacked the mRNA, this message where they were saying build a bicep muscle protein. Go to the next slide. Now what we want you to do is make a COVID spike protein. So they take that same message and they take a piece out and they stick a new piece in. They put the blueprint for the COVID spike protein. Now it says, dear ribosome, please make a COVID spike protein. And to make the COVID spike protein, do the following. Step 1, do this, do that, do something else. And when you're done, please drop it into the bloodstream where they know that this COVID spike protein will be in your blood system. The immune system will respond and will be able to attack it and will be ready for any time that you encounter an actual COVID spike protein. That's what they did. They took out the piece that had the blueprint for a bicep muscle and they stuck in there a COVID spike protein, the code for it. It wasn't alive. It wasn't a live virus that they're putting in, but your body then builds a COVID spike protein, puts it into your blood. Rather than address it to a place in the muscle, it said to put it into the bloodstream. That way the body and the immune system could respond. So let's talk about message for a moment. Messenger RNA refers to exchange of information from sender to the receiver. We were just talking about having tacit information, and you have to have a medium which to exchange that information. So it's the exchange of information requires a medium between entities for passing the information. Less obvious is the need for encoding tacit information known only to the sender across that medium such that the receiver may decode it by using that same protocol. English language is something we're all familiar with. We can decode that message back into tacit information so that the receiver can understand it and respond appropriately. Now, think about this. Under Darwinian evolutionary theory, there's a primordial life or a primordial pond that is nothing but a primordial soup. It's got chemicals in there. I love Jim Tour, who is a cohort and friend of Stephen Meyer, who is a synthetic chemist. And he is animate and vocal about this kind of stuff, that it's not possible for you to take chemicals in a pond and charge them with electricity and make life out of it. Now, they had a Urey Miller experiment. Some of you probably heard of this in 1952, I think, where they took bottled, purified chemicals, they stuck them into a test tube, ran electricity through it, and eureka, they were able to build amino acids. The problem is these amino acids are in a very, very pure form, and they decay over the weekend. So how do they get life? Amino acids are equivalent to words. And information doesn't get passed by words. Information gets passed by amino acid building blocks of life being written in sentence form. You have to give a blueprint for life, and then you have to, what Stephen Meyer will call, or Jim Tour will call, you have to boot it up. Go to the next slide. When they build these chemicals in a laboratory, when they put these chemicals together in a laboratory, they never not. They're always left and right-handed molecules. And there's an example of what they call left and right-handed molecules. They have an equal number of molecules, and they always come out in equal number, one that is called a left hand and one that is a right hand. That's the way they look. When life synthesizes proteins, it always only has the left hand. So one of the things that chemists have to do when they buy these from the chemical companies in a bottle, not only is it in the most purified form that you can possibly get, but they've taken all the right-handed molecules away and you only have the left-handed molecules. And when they finally build an amino acid, if they aren't able to replicate it and sustain it and keep it going over the weekend, when they come into the lab on Monday morning, they have to start it all over again. And what Jim Tour will tell you is if this happened in this amino acid pond, this primordial soup, we're supposed to believe that all of this purified stuff and only left-handed chemistry happened under a rock, and it lasted for 400 million years. And what he'll tell you very quickly is when you find a single mistake, those of us that have worked with computer programming knows, if you put a comma, a semicolon, you put the wrong character in the wrong place, somebody's laughing over here, somebody's gone through this, you're going to start all over again because it's not going to compile and your code is not going to work. And what they know in biology is you have to go back to the beginning. There's a whole process in the laboratory of steps. And the way Jim Tour explains this, you're baking a cake. You don't put flour and sugar and vanilla and frosting all in the cake at the same time. There's an order that you have to do it. The frosting comes after the cake has been baked. And what he has to do is freeze at super high, low temperatures, some steps, and super high temperatures at others. There's a step, there's an order. And if it happens for 400 million years and all of a sudden you find it's broke, you have to go back to step one and start all over again. And molecules don't have a mind. Molecules don't tend toward life. They don't know what life is. They're inanimate, inert material. They don't tend toward life. They have no reason to tend toward life. They're just chemicals. Without something intelligent forming them, for instance, Urey Miller in a lab in 1952, putting them together in super purified form with only left-handed molecules under the right conditions, and some of the amino acids that they created aren't life-sustaining amino acids at all. They created something, but they're not useful, they're not functional for life. So I've got a couple of videos that I wanted to watch. These are about 15 minutes long. We're going to look first at DNA replication. Go ahead and run that, please.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
So how many, Leon, how many protein machines did y'all count? Probably 5. I think I counted 5 in a very, very complex, particularly when they were going from the 3' to 5'. Don't ask me what that means. It's smarter than me. But they can only go in one direction, and so when it's coming down one direction, it can go from 5 to 3 in a continuous strand and just copy that DNA all along the way. But when it goes the other direction, it has to put a primer, and then it has to put that Okazaki fragment in there, then another primer, and then another fragment, another primer, another fragment, then another machine comes in. And does that sound complex? Do you think maybe something with pure chemicals that are mindless came up with that process? It is a very coordinated effort between 5 different motor machines, basically nano machines, that are doing this inside of your body billions of times every day. So we saw what Moderna said about the mRNA. Let's look at DNA making proteins.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Does that sound a little bit like what Moderna was describing? Anybody ever heard of this stuff? It's incredible what's going on billions of times in our body. Those proteins fold in a very particular way. Dr. Robert Carter is a marine biologist that does genetic engineering as well. He says that these proteins can fold in various patterns. In one way it'll form a particular protein, and when it folds in a different direction or in a different way, in a different three-dimensional fold, it'll produce something else. This is happening billions of times in our bodies. What was the next one there? The kinesin
Cells are full of specialized components that perform functions vital to their existence. But how do these components get to the right locations inside the cell to perform their functions? For larger components, a transportation system is needed. Meet the kinesin. Masterpieces of microengineering, kinesins are miniature motorized machines that carry cargo from one part of the cell to another, walking along self-assembling highways called microtubules. Known as the workhorses of the cell, kinesins have 2 feet, or globular heads, that literally walk 1 foot over another along the microtubule, pulling their cargo to its destination. Each foot possesses 2 special locations, called binding sites, that interact with other molecules. One site attaches to the microtubule, and the other binds with ATP, the energy molecule of the cell. When one foot binds with ATP and uses its energy, the foot flips over, resulting in the walking motion. Each foot has a short neck, which is connected to a strand of a long coiled stalk. At the end of the stalk is a fan-shaped tail, which holds tightly to the cargo being transported. Kinesins can carry cargo that are many times their own size. Sometimes a kinesin is in danger of getting stuck on the microtubule highway because of blockages caused by other cellular components. To get around such obstacles, multiple motor proteins may be used to carry a single piece of cargo, together providing enough force to break free. Kinesins typically walk away from the center of the cell and toward the cell's periphery. The kinesin's 2 feet work together efficiently, with one foot holding fast to the microtubule, while the other releases itself and takes a step forward. This coordinated stepwise movement allows kinesin motors to walk as many as 100 steps per second, moving about 8 nanometers with each step. When not carrying cargo, kinesins can shift to energy-saving mode to conserve fuel until their next job. The kinesin plays a vital role in many cellular processes, not just transporting materials, but also aiding cell replication. The walking kinesin molecular machine, another example of intelligent design.
Anybody find that fascinating? So these, it's going to keep on going, isn't it? Anybody fascinated by the fact that you have a post office running in your body? Anybody fascinated that when one gets stuck, another one helps it out? If it gets stuck and it can't carry that load, that we can have multiples, kind of like the team of horses on the old Wells Fargo wagon. They back up and they help pull it. And all of these have to be narrated by somebody with a British accent, otherwise it's not smart, right? All right, we've got one more, and then we're going to start looking at some pretty cool imagery that we have.
Motile cilia and flagella are hair-like appendages of cells that generate fluid flow along tissues or allow cells to move through their environment. To better understand how these appendages generate motility, we flash froze sea urchin sperm cells while their flagella were actively beating, and then directly visualize the molecular machines inside the flagella through cryo-electron tomography. Thousands of motor proteins, called dyneins, are arranged regularly along the microtubule scaffold. Through classification, we identified several different functional states of the dyneins as they step along the microtubules. We demonstrate a detailed stepping mechanism. From the relaxed post-power stroke state, the dyneins release from the microtubule, swing forward, and rebind to the microtubule. During the power stroke, the cargo is pulled forward by the action of dynein. As long as ATP is present, dyneins continue to walk along the microtubule. However, if dyneins receive inhibitory signals, they can be kept in intermediate states that resemble the post-power stroke state, leaving them unable to generate motion. Our direct visualization of specific dynein conformations on either side of the flagella suggests a switch inhibition mechanism generates the flagellar beat. As one side is inhibited, the other side is active. In our model, regulatory signals result in inhibited dyneins on one side. This allows dyneins on the opposite side of the flagellum to walk along the microtubules. Molecular linkers resist this sliding, which causes the flagellum to bend in one direction. By periodically switching the side of dynein inhibition, the flagellum alternates the direction of bending, resulting in the typical whip-like motion. The propagation of dynein inhibition in different regions of the flagellar wave along the length of the flagellum leads to the characteristic beating motion. The dynein inhibition may occur in an all-or-none fashion, similar to an action potential, which could explain how cilia and flagella can switch their bending direction back and forth in less than 1/100th of a second and continue beating for days.
How's that for motor protein? So it's not just a chemical reaction that chemicals tighten up and relax and start to, these are actual motor machines that are causing the flagella to do the things that it's doing. But I think is that video, the next one the video is up? We've got one more. We're going to look at the flagellar motor. This thing fascinates
Many
me.
species of bacteria propel themselves through their environment by spinning helical motorized flagella. Rhodobacter cells have one flagellum each, whereas E. coli cells have multiple flagella that rotate in bundles. Each flagellum consists of a helical filament that is 20 nanometers wide and up to 15 microns long, and spins on the order of 100 times per second. These animations show a series of schematized and speculative models about how bacterial flagella might function and assemble. Just outside of the cell wall, the filament is connected to a flexible rotating hook. The filament, the hook, and a structure called the basal body, located below the cell surface, make up the three parts of the flagellum. The basal body consists of a rod and a series of rings embedded in the inner membrane, the peptidoglycan layer, and the outer membrane. Some of the rings make up the flagellar motor, which can be divided into two major parts: the stator, which is attached to the peptidoglycan layer, and as its name implies, remains stationary, and the rotor, which rotates. The motor derives its power from a proton gradient across the membrane. In this example, a high concentration of protons exists outside, and a low concentration exists inside the cell. The protons flow through the interface between two types of proteins, called mote A and mote B, that make up the stator. Mutational studies suggest that a conserved aspartic acid in mote B functions in proton conductance. Each stator contains two mote B proteins, and therefore also contains two of these important aspartic acids. Although the molecular mechanism of rotation is not known, one possible model describes protons moving through the channels in the stators and binding to the aspartic acid in the MOTB proteins. This binding causes a conformational change in MOTA proteins, resulting in the first power stroke that moves the rotor incrementally. At the end of the first power stroke, the two protons are released into the cytoplasm. The proton loss causes a second conformational change that drives the second power stroke, once again engaging the rotor. Although the mechanism for motor function is not yet certain, many details of flagellar assembly have been determined. Flagella begin their assembly with structures in the inner membrane. 26 subunits of an integral membrane protein called Fli-F come together in the plasma membrane to form the MS ring. The Fli-G proteins assemble under the MS ring. Fli-G, along with Fli-M and Fli-N proteins, make up the rotor. Flagellar proteins destined to be part of the extracellular portion of the flagellum are exported from the cell by a flagellum-specific export pathway and assembled at the center. Mote A and Mote B form the stationary part of the flagellar motor, the stator. Both are integral membrane proteins, but Mote B is also anchored to the rigid peptidoglycan layer, keeping the stator proteins fixed in place. The subunits of the rod portion of the rotor move up through the hollow cylinder in the assembly, and assisted by cap proteins, build up the rod in a proximal to distal fashion. Another set of rings, called L and P rings, are found in gram-negative bacteria such as E. coli. They penetrate the outer membrane, forming a bearing for the rod. As the rod cap is exposed outside the L ring, it dissociates and is replaced by a hook cap that guides the assembly of the hook proteins. After the hook is assembled, the hook cap dissociates, and a series of junction proteins assemble between the hook and future filaments. Finally, yet another cap is built, and filament proteins assemble. Like the rod and hook proteins, they travel through the hollow channel inside the filament to reach the distal end. The cap rotates, which causes the subunits to build in a helical fashion. A complete filament can consist of 20,000 to 30,000 subunits.
That happened by accident, right? So what we have here is an animation, just a stationary animation of what these things look like. Look at how complex that is. Pure left-handed chemicals under a rock in some primordial soup just happen to fall that way and cause. These are those single-cell, simple life forms that we always hear about. Surely we started from a single cell, simple life form, and it evolved into a monkey and evolved into you. You hit the next slide. What they do is they take these simple life forms with the flagella, they freeze them at somewhere in the neighborhood of 1,000,000 degrees centigrade. I don't even know how in the world you get to that cold. They freeze them, and then they're able to take imagery as best they can, like this. Hit the next one. There you've got a bacteria in the upper left with that flagella hanging off of it. By the way, that young lady in the last video said it turns at 100 rotations per minute. That was incorrect. It's 100,000 times per second, and it will stop in 1/4 of a turn and turn and go the opposite way at 100,000 RPM the other way. These things are amazing. I think there are 13 sections in the various ways that they're able to image us. Hit the next one. These, by the way, are taken from various scientific studies. I took them from different, because I told you before, everything's a lie until I prove it otherwise. So I've gone to Harvard, I've gone to other medical schools, Rice University has a bunch. This one came from where? Japan, I think. So go to the next one. This is astounding to me. Now, if these motor proteins in the body that are building your cells, that are replicating your DNA, you've got motor proteins that unzip. We got motor proteins that copy. You got motor proteins that input a primer. You got motor proteins that will make that Okazaki fragment. You got other proteins that will destroy the primers. And then you've got some that will, the ribosomes, that will actually build proteins. That's all happening inside of a single body. We all know how mammals and sexual reproduction occurs. This is fascinating to me, particularly in the context of evolution. The spermatozoa cell carries that male DNA, and I answered your question there, Terry. I got an answer to your question. She asked me last week the difference between fraternal twins and identical twins. The egg has 23 chromosomes in it, and then the male sperm has 23 chromosomes in it. Fraternal twins, you'll end up with 2 eggs in the uterus, and they will actually make their own separate fetus, turns into a baby, and you'll end up with fraternal twins. What happens in identical twins is that 23 chromosomes in the egg and the 23 chromosomes in the sperm fertilize the same egg, and for whatever reason that egg divides with the exact same identical chromosomes. So both of these fetuses, children, unborn, are being developed in the womb with the same, and from there the differences are less than 4 to 5%, they say, and most of the differences are due to environmental aspects. But that's why they look alike, and they're always the same gender. Anytime you have, I bet your brothers, you said, were identical, right? And they're both boys. What's happening in sexual reproduction? Let's think about squirrels or animals, anything but human, just to keep this up on the, we all know how birds and bees work. But think of a tadpole. That sperm isn't just flying around in the female aimlessly. What's happening is the egg emits, let's call it a pheromone. It has a chemical that emits in the environment, and on the nose or the face of that tadpole, on the face of that sperm cell, it actually has a detection system that is keyed in looking for that chemical so that it can find its way and go find the egg cell and be the first one there to fertilize the egg. Meanwhile, the flagellar motor on the back is moving around at 100,000 times per second, and it can guide and navigate itself to where it needs to go. Now, mind you, that this is a male and it's female that are separate. There's a psychological, we know what happens when your dog goes into heat. We know what it does to the male. There's a psychological aspect of this that causes them to attract. They are made for each other, let's say. And then when the spermatozoa enters that female, they are specifically keyed to attract one another and to fertilize one another. And these are 2 separate bodies that are happening. How does that happen? By accident. Knowing what we know now about the flagellar motor and about these chemical responses, that I'm not going to it's too smart for me. There's big city words up there. I can't do that. That's incredible. There's no way that that happened by accident. There's too much there. Go to the next slide. Again, you have Stephen Meyer. Biology is part of the information age. Inside the cell, we don't have chemical reactions going on. It's an information storage, transmission, and two different processing systems. One for DNA replication, one for construction of proteins. The sophistication of the information system that is at work inside even simple one-cell bacteria, I think we just saw that, have information processing and storage capacity. That's Stephen Meyer. He's the one that, again, wrote Darwin's Doubt and Signature in the Cell. I recommend both books. They're good stuff. Now, one of the contemporaries for Darwin, I'm almost out of time, I better pick up, was this guy. Lyell was his name. He was a geologist, and he came up with an older form of the scientific methodology. We all understand what scientific methodology looks like today. In the 1850s and 60s, when Darwin and this guy Lyell were operating, he said the answer for things that we see in geology, we ought to look at causes in operation now. What things do we see happening in the world today, right now today, cause the things that we see similar to in geology? So go to the next slide. So the question is, in biology, what is the cause now in operation for the production of digital information? How do we have biological information encoded with A, G, C, and T in your DNA, blueprints, so that other motor proteins that just happen to be there by accident can copy, replicate, and build additional proteins? Somewhere in the neighborhood of 156 proteins are required to work together to make a single protein. Go to the next slide there. So this guy is at Arkansas Tech University. He's a marketing professor, and he wanted to go check on all the people out there. And he asked a very particular question. He asked it two different ways. Go to the next slide. He asked the same question twice, and he just changed a couple of words that I have bolded there. So scientists researching origin of life have created complex life forms from scratch, out of a bottle, basically. That is, in a laboratory experiments that approximate Earth's early atmosphere, scientists have mixed chemicals believed to exist before life existed and successfully created complex life forms such as frogs. 41% believe that frogs came out of a lab after they mixed chemicals together. 41%, that's amazingly high. What's the basis for that? They've never had a report of that, but they get these reports out of scientific studies. They even put these, and Adam had shared the biology book that they have over at the high school for freshman biology, where they talk about evolution and whatnot. And they take these caricatures that is, I'll put it in air quotes, age appropriate, and they lead these kids and they lead these young adults to believe this kind of stuff. Go to the next slide. They ask the same question and they change complex to simple. The scientists have created simple life forms such as single-cell bacteria. That went up to 72%. Almost 3/4 of respondents believe that has actually happened. It has not. They have never created life. They've created a few amino acids. We talked about the Urey-Miller experiments in 1952. Jim Tour will tell you since 1952, origin of life science has not progressed one little bit since the Urey-Miller experiments. He is animate about this, and he says he's had scientific research paper after research paper sent to him, and he says, stop sending them to me. They all are in the same category. They mix a bunch, they glorify, and they overhype the finds that they actually had. We'll get to see this here in just a moment. I'm well behind it at the moment. But they're all the same. Go to the next slide. These are educated people 80% of them, I think it says, 80% hold college degrees. Almost 3/4 of those are at the bachelor's degree or higher. These are educated people, but they're getting this out of the current science. Go to the next slide. This guy, Jim Torr, will talk about in 2010. This guy and his team, after 15 years, announced, I'll just read it, he and the team made a historic announcement. That URL, by the way, at the top, you can get this off the church website if you want to go back and look at it later. Hopefully you can read it. Is a full press. He's standing at the podium and he's talking about the history and how they came to do the things that they did. And he claims that they got the first fully functioning, reproducing cell controlled by synthetic DNA. He explains how they did it and why the achievement marks the beginning of a new era. That's not hype, is it? That's a whole new era in science. Go to the next slide. Jim Torr is commenting on there. He says there's no rivalry with natural life here. He says all the active chemical components were extracted from living systems. These are the most, if these are the most lifelike artificial cells yet, this serves only to underscore the point that no one has ever come close to the real thing. One of the things that they do is they'll take these left-handed chemicals off the store shelves, they'll mix them, shoot lightning bolts through it, and out of this muck that it creates, what happens when you put sugar in a pot and start making caramel and you leave it too long? It burns, right? You have to stop the reactions. So in nature, there's nothing controlling it to stop the reaction. We're supposed to believe it stopped at caramel. So you've got this muck, and the backbone of DNA is ribose. It's a sugar, ribose. That's where you got ribonucleic acid from. They put all these chemicals into the pot, shoot lightning bolts through it, and they got one fractional piece of ribose. And they say, Eureka, we created ribose. And then they moved to step 2 by taking chemical ribose off the shelf from a chemical company in larger proportion, and then they moved to step 2. Literally, that's what they have done. And we're supposed to believe that in a primordial soup, absent of any intelligent design such as God, let's say, it did this all by itself. Go to the next slide. This is Jim Torr again. I love this guy. I totally recommend you check him out. How is molecular machinery made? They do not say the mechanisms needed for cellular division. He's still commenting on Craig Vanier, the last guy we were looking at. The mechanisms needed for cellular division and complex requiring cascades of precisely functioning enzymes. Enzymes are those motor proteins. There is nothing between what the authors demonstrate and what they claim to have established, and nothing they propose, quote unquote, paves the way for the first living microbial community. It's part of this press conference that he had with the full court press in front of him. He just says, on such and such a day at 6 a.m., Danny called me with a text, we've created a blue colony. Exactly what he says. Eureka, we finally created life. Go to the next slide. He says, this is what they've done. He says, I buy a Corvette and I take the computer console out. I take it to the university, put it on the lab. I pull the chips out. I made a few chips on my own. I take my chips, stick them in the computer console. I take them back to the Corvette, stick them in, slam the hood, start the car. I created a Corvette. That's what they did. They took pieces out of the cell, and then they go put other pieces back in the cell that they've modified. At one point in 2016, this same guy, in the spirit of what Michael Behe, you remember Michael Behe's mousetrap, irreducibly complex. How many pieces can we take out and it still function? And they got down to 473 parts. They took out one more and it failed to function as a cell. And he says, Eureka, we have created a protocell. And Jim Torr says, no, you didn't. You made it worse. You took a functioning cell and you made it bad. Because you took out all the good stuff that was in it and you figured out what makes a cell, we think, or they think, function at the lowest level, the most irreducibly complex before it fails to function as a cell. All right, that's everything I have for tonight. If we are not a product of God's design, either something's wrong with you or something's wrong with me. I'm telling you what we have seen tonight, and you culminate that with what we've seen over the last 8-9 weeks. We are see what I mean? We are irreducibly complex. We are interconnected, interdependent, because the brain doesn't function without the heart, and the heart doesn't nourish the brain without a circulatory system with blood running through it that just happens to take oxygen out of the atmosphere, without nerve endings to connect them, that the heart is irreducibly complex, just as a machine by itself. It has valves, it has myoelectronic connections, it's got pulmonary connections. And now we see, when you go down, what makes the body decide it needs a heart in the first place. Why build a heart in the first place? Molecules don't have a brain. They don't tend toward life. I just said that, right? But somehow they decided they needed a heart, they needed a brain, and they needed all these myoelectronic and circulatory connections to function. And that supposedly happened by accident over millions and billions of years. I'm not buying that. It's been said before, I don't have enough faith to be an atheist. Questions and comments? Oh Lord, here it comes. Everyone's dumbstruck now. No volunteers. Well, perfect, because we're right at 7 o'clock, so I can close us out. Nobody? So, what Scott said is it was intellectual design. Dr. Meyer, I'm going to tell you what, Dr. Jim Tour, is a professor of synthetic chemistry at Rice University. If you go to Rice University's website and go to his page, in a drop of a hat, he's got his contact information there. If you are not a believer in Jesus Christ, please contact Steve Tour. He will set up a Zoom session with you, and he will take you through. Now, when he gives conferences, at the very beginning, he says, yep, I'm a Christian, I believe this, I'm going to purposely take God out of it because I want to show you what we just went through. Now you decide if that happened by accident. I'm telling you, go look up Jim Tour. He's a wonderful, wonderful guy to hear, and he's very animated. I think he's from New Jersey, so all of them from New Jersey are animated, right? Anything else? Once, twice, it's 7:06. I'll close this out in prayer. Father, thank you so much. Just for the ability to live in an age where we can see things that past generations could not see. They could see things from nature that we can see so much more now. It is so obvious that it was by your power and your majesty and all for your glory that we were given that neshama, that breath of life, that all creatures, big and small, the whole variety of life, the species of life, we can actually tell, just looking at common science, how these things happen. We can see your glory shine through all of that, and we thank you for that. Father, until we come together next week in our final session, I ask you would put a hedge of protection around each one of us and bring us back safe next Sunday. I ask in Jesus' name.